This is a production of Cornell University.
Thank you very much for the invitation. It's a real honor to be here and to speak, and the chance to tell you about our work. And I will jump in so I don't run over time and give you an overview on some of the really interesting questions we've been able to ask in fungal evolution. And so I want to also pay tribute to the beauty of the different organisms that we get to work on in fungal biology and in mycology. This is sort of just a couple of plates from a recent paper book chapter that we put out that is part of this new book called The Fungal Kingdom, where there's a lot of really great chapters about different aspects of fungal biology, but it includes some paying homage to some of these beautiful structures. And I think what I want to emphasize is that if you don't work on fungi, is that they are, of course, many different cell types and shapes and sizes and colors that we see, and trophic interactions. And so trying to understand the evolution of these groups of organisms, how they're related to each other, and how some of these different forms came about is one of the driving questions that we've been doing in my lab and across the collaborative community in this work. And so one of the driving questions is what are the evolutionary relationships of the fungi? If I minimize this, that's okay? Okay. So what are the evolutionary relationships among fungi? This has been a driving part of fungal systematics, first, we used morphological characteristics, sometimes ecologies to group things, then with the ability to use molecular systematics to resolve the fungal phylogenies. That was driven in part by the ability to use PCR to sequence the ribosomal locus regions. And then one of the kind of landmark papers in 2006 as part of the Tree of Life work was able to use a 6-gene phylogeny to resolve the major lineages. And we thought, okay, we've got a good map of how these organisms are related. But in fact, as we start to look carefully at those trees, we realize there are places that we don't really fully understand the resolution. And so we need additional sampling of both markers and lineages to get at that. And some of the places that I'm going to emphasize where we don't know the answer, and we maybe still have questions, relate to some of what we might call early branches, or at least what are the less appreciated lineages in the past, perhaps, but things like ZYGOMYCETE fungi and the flagellated chytrid fungi, where some of the order of the branchings have been unresolved, and that even within groups of things that are well studied, like the Basidiomycetes, which include mushrooms, but also smuts and rust, some of those relationships unresolved, even with sort of large sampling and gene sets. So one approach to try to solve this problem is to sequence more. One question that we'd like to understand is how they're related, but why do we care about the tree? So being able to have a tree of how they're related to each other allows us to look at when characters evolved. When did ecological associations arise? Did they arise multiple times? Not all plant pathogens come from a single origin, so timing when those changes occurred. But also when we look at more broader context, the rise of diversity of fungi could be correlated with the rise in diversity of plants, or rise in diversity of insects. And so understanding better the timing is what the phylogenetic work allows us to present, and then to correlate that with other kinds of characteristics. On top of that, we also can see things like interesting morphological origins. And so, for example, on the top left here, I will only briefly talk about this fungus called endogeny, which looks like a truffle, but is part of the zygomycete fungi, which are typically, we look at them as filamentous molds. And so we have examples of things that have really complex morphology embedded within a group of organisms that don't share that. And so understanding how many times those kinds of complexities arise is really interesting and requires a well-resolved phylogenetic tree and relationships. So how do we get at that? We try to sequence lots of things to get at this better, right? This is probably an overused graphic, but the idea that we would like to understand how they're related, the easiest thing that we can do, especially with today's technology, is just turn the crank and generate more sequencing. And so, of course, through our partnership with the Joint Genome Institute, where there's a lot of sequencing capacity and the interest and ability to try to generate more sequencing, more genomes, and sampling the lineages to capture this diversity. And so our effort that with the initial, what we call community sequencing project that we proposed was we called it the 1,000 fungal genomes because we wanted to have a big, large number. And there were at the time approximately 500 families of fungi. So this is the Noah's Ark of fungi. We get 2 from every group and we make sure they're covered. Truthfully, there are, of course, more than that, but more families than 500, but this is a nice starting place. And so the initial goals are to better reconstruct the phylogeny using many gene phylogenies, not a 6-gene phylogeny, but also to look at how the genomes evolve, look at how they change in content, look at things like how carbohydrate active enzymes have changed, which relate to how organisms get their nutrients, but also other aspects of their biology. So I'm going to give you a survey of some of the things that we've done. I'm going to indulge myself a little bit and talk a little bit about some side projects that I think are interesting that were related to some of these investigations, and then tell you one story about complex multicellularity. So another part of this is, of course, it's important to take sometimes a focused approach to some of these questions. And so one group that's not received as much attention in the past for some of these genome sequencing projects and molecular genetics in some ways has been what might be called the early diverging fungi, but the zygomycetes and also the chytrid fungi. And so zygomycetes were previously thought to be one lineage, but I'll show you in a second that there are actually 2 major lineages of that constitute what are called zygomycetes. And our funded project to look at these was to target about 150 reference genomes in this lineage, these lineages, and also to generate between 500 and 1,000 light coverage genomes that allow us to fill in the gaps. So I just want to take a second. Okay, we live in 2017. Even when we proposed this, it was 2014. But that we could take a really understudied group of organisms and propose to do 150 genomes in that. It's kind of staggering to me to think about where we are in our lifetime, in our technology that we have available to, and the costs. So I can't project yet what's going to be 10 years from now, how straightforward, hopefully, some of these things are going to be, and the scale of things that we can do. So I want to take, sometimes we're flippant about how many genomes we can sequence, but I do recognize that this is a nice thing that we have access to now, in a way to drive our questions by sequencing. So in addition to sequencing, we also want to understand the biology of these organisms, and previous work had been looked at aspects of the characteristics of these. So for example, fungi that grow in hyphal forms have a concentration of vesicles at the tip called the Spitzenkorper, and that's found in dicaria fungi like Aspergillus or Neurospora, and it's found in some of these zygomycete fungi which form hyphae, but not all of them. And so Robbie Robertson, who's an expert in imaging, has been working on using this big collection of zygomycete fungi to look at which of these have Spitzenkorpers and when that evolves. That's one side question that's being looked at. A second thing is that these are typically, if you teach mycology, you take mycology, we talk about zygomycete fungi as senocytic, is that they don't have septa. But in fact, that is not the case for all of these. And so understanding when and how septal compartmentalization evolves within these groups is important. And the other thing, just to show you, we don't know everything about these fungi. When you have better imaging, I had a movie, but it's not going to show here, but these little vesicles here, we don't know what they are. They're probably lipid containing, but they actually move quite fast. I have a real-time video and they're just moving quite fast. And this is a Mordiarella species that it makes lots of lipids. Okay, and so understanding this is useful in the industry, but also interesting from a biological standpoint. And then the last thing is just that it's a technological project, and so some of the efforts to generate single-cell genomes, because some of these organisms we can't grow alone, and so we have to grow them with a host. And so some of the projects with the JGI are about taking infected tissue and isolating single spores or single cells, sequencing those, and so we have several single-cell genome projects that are going on. Excuse me. Okay, so what can we do with some of these genomes? I'm going to give you some 10,000-foot view views of these data. And so, for example, these genomes vary in size right now, and this sort of sampling that I'm showing you between, excuse me, 2 to 150 megabases in size. Now, there are fungal genomes that are about 700 megabases to about 1.5 gigabases. These are some of these entomopathogenic fungi. They're harder to sequence because they're so big, but we do have draft assemblies of a few of those. And so there are some things that are bigger than that. They're full of transposons. But here's a snapshot of the main bulk of about 400 of these genomes that have come out. And what you can see is that as genome size gets bigger, you have more genes. But you do have species that fall off that curve that have large genomes but not so many genes. And those represent genomes that have a lot of transposable elements in their genome. So the genome is bigger, but that space is taken up with by transposons, not protein coding genes. So that's interesting, observationally. And we can also look at, hopefully the next page, we can also look at another dynamic of that, maybe, of genome size and gene size. So for example, some of the organisms have very small genomes. Microsporidia are these obligate intracellular parasites. They have very small genomes at the bottom left corner here. 2 megabase, 4 megabase genomes, and but they also have very short genes on average. So you've made your genome smaller, and the genes in your genome have gotten smaller. And so that's an example of presumably of contraction. And then you have other organisms that have long transcripts, but not necessarily a bigger genome up at the top here. So there's different patterns. Not everything can be ascribed to ecology, but those are interesting observations we can make. And then one other thing that we can look at is just the genome, the size of the genome and the number of introns that we see. And so this is a case where we can see different clades. So the colors here represent different major clades of fungi, that different clades have really different properties. And this is, again, indulgence to my PhD thesis where I worked a little bit on this. And what we find is that things like Saccharomycotina, so Saccharomyces, have very few introns. That's that top left corner here, right? So they only have like 300 introns in the whole genome in some of these species. And then we have other species down here at the bottom, the mushrooms, that tend to have many introns per gene. Okay, and so the scale is kind of reversed because it's about how big the exons are, not how many introns. But the idea is that, so you can see that this kind of falls out by some of the colors. And so there's a history in how gene structure evolves in fungi, and we're able to compare that across common sets. So anyways, there's differences. Fungal genomes are not uniform. That might be one takeaway that you can get from this. And some of that's driven by historical patterns. Okay, so how do we understand how these organisms are related to each other? So one thing we can do is try to build phylogenetic trees. And so one pipeline that we use is using both a set of predetermined conserved proteins that we have 434 that we've picked out that behave well as being found in most fungi and also in some of the outgroups that we were going to use. And we identify these proteins in all of our genomes in a rapid succession, and then build multiple alignments of each of those. We trim and quality control and concatenate those and build phylogenetic trees from concatenated data sets as well as individual gene trees. We look at congruence of all of these and try to generate a consensus phylogeny of the organisms to understand how they're related. So for those, again, this is maybe for those of you who are interested in the methods part. And then we're also trying to understand when these organisms diverge from each other. So we also incorporate fossil data that's been collected and calibrate some of the time points in the tree. Okay, and when I first started this project, I was like, oh, fungi don't leave fossils. What do you mean? But Tom Taylor, the late Tom Taylor, in his work, did amazing work in paleobotany and being able to collect and identify and characterize structures that seem to be fungal. And he was part of our grant and our project to collect more of these, especially for the zygomycete fungi, but also to put that in. So again, I've already showed you this tree. We can generate a really big, beautiful tree that is hard to read, and then we can summarize that into the other major lineages. And so some of the things that we learned, and I'm going to come back to this point again, is that these are sort of known relationships of the Dicaria fungi, the ascomycetes and basidiomycetes, but that one of the major findings is that what we call the Mucoromycota now represents these three lineages. Used to be kind of arrayed more closely with the Dicaria, that this Glomeromycota used to be more closely related here, and now we find them as one group, and that the second group we call Zoopagomycota represent organisms that are entomopathogenic fungi, as well as mycoparasites that infect other fungi. So those are really fun to study, but hard to dissect in the lab. And then we have some unresolved lineages in here that represent lineages of the Chytridiomycota, which we have the chytrids and the Blastocladomycota, both of these groups have flagellated zoosporic fungi, but the branching order has been a little bit ambiguous. We do think that probably Blastocladomycota is the earliest branch, is sister to the rest, and that the Chytridiomycota are sister to these, but that's been still a little bit challenging. And again, there's other unknowns that we have that I can just throw in here more for the aficionados that help us try to, we have Basidiobulus and Opidium, which are part of this looks like a chytrid, but it actually groups within these groups right now. And so trying to understand these relationships is still part of things that are ongoing. Okay, so how do we look at the dating of this phylogeny? By incorporating these different fossil calibration points, we're able to put some dates on the fungal emergence. And so one of the interesting things is there's a window of time when many of the fungal phyla emerge, which is also related to a similar timing to when we start to see the explosion of land plant diversity. So there's, but what's interesting is that a lot of these are kind of timed to the same, within the same period, and that that a lot of the diversity that we see kind of arose here in this window of time as best we can estimate. So again, these are helpful in trying to put the context as to when did fungal diversity come about. And again, if you hadn't seen this before, but we do think that kind of the origin of fungi, depending on how you define fungi, but is probably in about a billion years ago. So that's an old group of organisms if you put them all in that. The way I say if, because there's some debate or some worth of discussion about how do you define what is a fungus, and there's actually a great chapter from Tom Richards in that book about these definitions. But at any rate, this is helpful to look at the context. So what can we do when we look at these genomes of these organisms? And so one aspect that is interesting is we can look at gene diversity and innovation and duplication. So what I'm plotting here is a species tree of about 72 fungi, and I'm putting in these colors these circles where there are pulses of more copies of gene families. And so the idea is this is when there has been innovation in the history of these organisms. And so you can see that there are a couple of, so it's not uniform, right? It's not that there's a pulse at every node. There are certain nodes that have a lot of gains. And so these seem to be related to this big burst of diversity. And so one of the things, again, when we talk about when did fungi emerge, at this label I put here at the bottom, there isn't a lot of diversification happening at that node. It actually happens a little bit later in terms of the gains that we see in terms of new gene families and new diversification. But that's another way that we can kind of put this in context. And so we do think this helps us better understand when some of the changes that might have driven some of the fungal specific biology might have occurred. And we also do find big examples of gains, and I'm going to talk more about this group in a second, that relate to horizontal gene transfer as well. Okay, so that one group I was just mentioning, and I'm going to go through some of this a little bit quicker so I can get to the end, but is the NEOCALLOMASTIGOMYCOTA fungi, which are really fun to say 10 times fast in a row, but are the anaerobic gut fungi. So you can find these we're on an ag campus, so you might have cows that have fistulas. You can go in there and you can sample fluid from the rumen, and inside that anaerobic fluid, you can culture anaerobic fungi. And these are zoosporic fungi that are really weird. They don't have mitochondria, they have hydrosomes, and they are obligately anaerobic. And so in a project that we're working on with Mustafa El-Shahid and Noha Youssef at Oklahoma State is to sequence transcriptomes, because these genomes are a little bit hard to sequence. They're pretty repeat heavy. We sequence transcriptomes of about 20 to 25 of these species to try to better understand their relationships. And so, and this is a tree showing the dating of this, which is interesting in this context because this group of anaerobic gut fungi, these are all the lineages that we can find. We're able to sample of these groups, and that they emerge relatively recently. They actually emerge pretty close to, that's what this green box here is, the origin of herbivory in mammals. So the ecological niche where we isolate them from correlates in time with the age of the group. Now there's a whole lot of time on this branch between that. Where did they come from before there were rumen guts to live in? I don't know, but this is what we can talk about with the samples that we can sample today. So there's maybe other interesting diversity to look at. but this is an interesting way to look at that. And then one other thing we can look at is the evolution of some of the different structures. And so, for example, some of these have polycentric and monocentric forms. And so we can see again that those probably emerge multiple times within these lineages. And so again, being able to look at now gene content and genes that are found in these groups is part of trying to understand the diversification there. And we can even find genes. This is just a kind of a pictorial, just this is just to remind me to tell you that these fungi have lost certain processes and gained other things. And so here's an example of a gene that's not found in any other fungi except for some of the except for one weird group called Basidiobulus, but that they have gained genes that an example of a gene that is probably related to its growth and its anaerobic growth. And so we can find things that probably they had to pick up. And the reason why I'm showing you all these branches is that all of these organisms, all of these AGF fungi have it. So it isn't a horizontal transfer of just one or a bad genome assembly where we just picked it up accidentally. It's actually found in all of these lineages, and it's diversified, probably following the species diversification. So these might be genes that are necessary or important in that adaptation to being to that ecological niche. So we think this will be cool ways to better understand the biology as we start to map these. And there aren't that many genes that fall into these categories of being unique to or maybe missing from these anaerobic ones, but we do see again like mitochondria lost and things like that. Okay, so another thing we can do when we have these genomes is try to ask other questions about genome evolution. And one interesting thing that people ask a lot in plants, but hasn't been asked as much in fungi, is what happens when you get a genome duplication. Now, there's a well-told story about Saccharomyces cerevisiae. There was a whole genome duplication about 100 million years ago, and we can map what happens to gene duplicates in the time that occurs after the duplication. And this is really cool for evolutionary biologists because there's lots of theory about what happens when you duplicate genes and you see what happens after that. And in plants, one of the ways that this was looked at from transcriptomes, in fact, and something from Ken Wolf's lab that was first published were these graphs where you show a plot of the average synonymous site differences. So synonymous sites are basically supposed to be neutral changes that you can count. Basically, they're calibrated with time. So if you have a whole genome duplication and you had 100 genes and you duplicate that to 200 genes, and each of those has a pair, okay? And at time point 0, right after the duplication, the difference between any of those pairs is 0 because they're identical. But as time passes and independent mutations occur in those gene pairs, each of them starts to get different from each other. And that difference we can measure with the synonymous site difference. And so that difference looks kind of like a peak. It's not perfect, right? Not every gene gets the same number of changes, but that peak starts to move over time. And so you can actually guess when the duplication occurred by looking at the distribution of this peak. Right? And in plant genomes, when we've seen multiple duplications, you see one peak, and then you see actually another peak for multiple duplications. There's like an echo, okay? So that's the model, that's the way you can approach this. So we can do this. We have 300 genomes. We can look for paralogous genes in those genomes, and we can ask, what's the synonymous site divergence of them? Okay, and we can find examples like this for a fungus like a chytrid fungus called Allomyces, which has this nice pattern of lots of things that are duplicated, right? This is the sort of distribution that we see. And it looks like it's probably actually a diploid assembly, but the alleles are pretty different from each other, so they didn't collapse into one. So I don't know the actual biology of why it has so many copies, but this is an example where we think it's probably because it's a diploid. And we can look at something like this across all of the genomes, and we get these patterns, right? So nothing to look at specifically there, but we can pull up something like Saccharomyces cerevisiae, where the duplication occurred 100 million years ago. So that KS value has gotten really big. There are not that many pairs that are that similar to each other, right? This one has this about 0.5 for the Saccharomyces. Again, there's only a few that are really similar. Everything is kind of enough time has occurred, okay? But we can look more carefully, and we can actually find an organism that does seem to have this whole genome duplication that occurred quite recently. And that's a fungus called Hortea werneckii. And this had been previously described, so I'm cherry picking here. This had already been known as a genome with whole genome duplication. And they had done an initial kind of low quality Illumina assembly, and we did a PacBio assembly in collaboration with Corey Nislo's lab in Canada. And we're able to then look at the genome duplications and identify that most genes were in this orange category here were duplicated. So most of the genes in the genome had 2 copies. And then we can look at the dates of those genes, the average age of those, and see that divergence time. And so what's cool about this is probably this is really a real whole genome duplication, and it's probably the most recent one that we can identify based on these comparisons of all the genomes that we have. Because there's so many genes in the genome that are basically one-to-one paralogs, and that we can map it on. And so this is what it looks like. It's a black yeast. It's an extremophile in the sense that it grows in really high salt concentrations. It was isolated originally from these salterns. When you make sea salts and you let the water evaporate, you can find some fungi growing in this really high salt molarity. You could also find it in marine environments. And it's kind of cool because they call it a yeast, but actually it grows filamentously. It's a diphidiomycete. And so if you look kind of at the, you look at these cells under a dissecting scope, you can see that they're actually these colonies that have hyphae coming out. So they're not really yeast, even though they make these kind of gooey black stuff here when you look on the plate. And this is, again, this is my other indulgence, is that in our lab, we've also started working on some extremophiles. And so I had a collaborating visiting student who works on Antarctic endolithic fungi, a whole other interesting question of, you break open rocks from Antarctica and you culture what's inside them and you think nothing. But in fact, there are a bunch of fungi that do grow slowly. And so one species of that is called Hortea, and a species called Hortea thailandica, which is actually isolated from Thailand. So it's not only an extremophile living in cold, but this is an isolate that came from Antarctica. And we sequenced the genome of that and put it in the tree, and it's the closest thing we have to it, but it does not have the whole genome duplication. So by being able to put this in the phylogenetic context, I can tell you that there's a species that doesn't have the duplication and a species that does. So now we have the ability to look at a single copy gene in one species that's duplicated into two in another. Okay, so this, again, from an evolutionary molecular evolution perspective, this is a nice toolkit to be able to explore more about that. I don't know that any of this tells us why it's able to live in Antarctica or live in these extreme environments. But again, just an advertisement, we've sequenced in blue or other species from Antarctica that we sequenced genomes for, and we're trying to understand a little bit better how they, are there any things about being an extremophile that are diagnostic for that? One other thing about these duplicates is that we can look at not only just the genes that are there, the copies of the genes, we can look at their expression. And so the gene expression differences on the left here in blue are how different the expression values are for these gene pairs. And so in general, they tend to be very similar to each other because they just duplicated. And they tend to have sort of the same nucleosome occupancy, but there is some variation. This is comparing something like Saccharomyces, which has had a whole genome duplication, which is a lot older. And so you see more variation in the kinds of differential expression that you're going to see about copies. But there are examples in Hortea where there's a gene copy that's expressed in high salt environments and a copy that's expressed in low salt environments. There's transporters and there's other things about adaptation to that environment. So there's more to it than this, but this is kind of one snapshot view of looking at it at the ecology of an organism that's had this duplication. All right, so I'm just gonna probably not going to cover everything today, which is fine. But as the ZygoLife project is, again, this ability to sequence lots of these genomes, trying to sequence both with low coverage genome approaches to get low sampling, use low amounts of genome sequencing to still get at enough information to build gene trees or species trees and try to do that. Can we do a better job in sequencing the ribosomal locus for doing barcoding? And so that's part of the driving that. I'm not going to put the trees in here for this, but it is working well. But we can also try to study it. And so again, this is this improved tree of the zygomycete fungi that are really in these two major lineages, the Mucormycota and the Zoopagomycota. Again, this is just annoying for those of you who teach mycology because you got to update your slides. I just want to advertise this is important in trying to understand the biology there. And that again. As I was saying, the Glomeromycota or Mycotina, do you feel like you've been demoted if you have a Mycotina versus a Mycota? But anyways, these are, again, important parts of that. But one other thing that we've done is that the Glomeromycotina are an important lineage. It's hard to sequence these. These are obligate. We can't sequence them. We can't grow them, that many of them in the lab. So through single-cell RNA-seq, single-cell amplification and RNA-seq, we're able to generate transcriptomes with work from Nico Karati's lab. And and this just came out, this finally just got published this week, but we have improvements on the sampling of the phylogeny of the Glomeromycotina using this single-celled RNA-seq approach. So again, improvements to the phylogeny helps us better build a backbone that's based on many, many genes. So now this is a 400 or 500 gene-based tree. Okay, so these are, again, just approaches that we're able to use, and this isn't so important, but just that pretty pictures of these different cell types, and that we're able to grow. We had to isolate these all from a plant and then sort the cells and sequence from there. And really, this is all pioneered by Nico Karati's lab in Ottawa. Okay, so the last thing I'm going to do in my 10 minutes, you want me to stop it?
No.
Okay, I'm looking 15. Okay, I won't overwhelm you. But is this one last question about evolution, multicellularity, and fungi? So this is what I've been interested in. I've shown you lots of pictures of microscopic fungi. You, of course, hopefully you live in a beautiful part of the world where there are lots of mushrooms. You walk outside and you see a mushroom. Or if you're unlucky and you leave, and where I am, you leave the hose on, and suddenly there's just this weird circula growing out of the ground in your water, in your flower bed. So some fungi you like, some fungi you don't. But the point is that you know that there's really different morphologies and a really different complexity that we might see in some of these. And so what we'd like to understand is how did some of that come about, and can we map when in time, and what kind of changes might be important to that. And so we did this in part because we observed these examples, multiple examples in the fungal tree of organisms that make multicellular structures. And so here I'm showing you sort of a sampling of the fungal phylogeny. And here I'm showing you this thing called endogeny, which falls within our zygomycete fungi. Right, so most of the zygomycete fungi are sort of filamentous. We have this thing that makes a truffle that looks something like a tuber truffle here, and then we of course have mushrooms, and we have some other examples of structures. And one of those is an organism called NEOLECTA. It's in blue here. So NEOLECTA falls within a group that you might recognize at least one of these names, Schizosaccharomyces palmi. Right, a 4,500 gene yeast, or that single celled that has this fission yeast. And then we have this organism called NEOLECTA that makes these fruiting bodies that look a lot like earth tongues, but they're not. And so this has been an enigma as to what this thing is. First of all, is it really an ascomycete fungus? Is it really an ascomycete fungus that falls within what we call the Diphragmomycotina? Pneumocystis is like, again, like a single cell obligate parasite of lungs. Right? We have things in here that this Cytella is a yeast. So why are we seeing this sort of thing that makes these big, relatively big structures? How did that come about? So again, if you have a hammer, everything is a nail, so we sequence the genome of it, of course. Now this is we can't culture it, so we had to sequence from fruiting bodies, which worked out okay. It made a reasonably good assembly, but not as good as we get with some of the other projects. But it allows us to try to use this system to try to compare NEOLECTA with these multicellular fruiting bodies to other organisms related to it that don't make that, as well as things that we have good model systems for, like Neurospora crassa or Aspergillus nigilans, or Coprinopsis, the mushrooms. And so we can take those spore carps and sequence them. And so when we look at some really, again, like top-level statistics, are genomes of organisms that are multicellular more complex in the sense that they're bigger, right? You might think that if you have a more complicated fruiting body structure, maybe you have more genes. And the answer is no. So here we go, NEOLECTA falls within. All of these are basically what we might call single-celled yeast, mostly yeast forming for the most part. And here are some of our filamentous Physizomycotina fungi. Here are mushrooms. And you can see that it does not have more genes than its relatives or the other things that it share that morphology. So it's not a gene count perspective. We can look and see if there's any example of lots of gene gains. We just have a lot of novel genes that nobody else has. And NEOLECTA is in blue here, and basically we only find this sort of number, something on the order of this sort of 800 gained relative to some of its other relatives, or on this branch, only about 25 genes really represent sort of a novelty in this lineage. So it's not gaining a whole bunch of stuff and losing a bunch of other stuff. It's not like it has some massively new set of things. Okay, so that's curious. That was maybe one believable hypothesis or plausible hypothesis that was not supported. So then we wanted to look more broadly. What are the genes that we can find in other organisms that we would call complex multicellular? And some of these organisms make septated hyphae, and they have channels to separate cells, which are really different. We wanted to understand, when did this gating mechanism evolve, and I'll explain that in a second. And then we also have mechanisms for how hyphae fuse together and remodel, which is really a major principle in how you form mushrooms, right? There's this whole knotting and fusing of hyphae to make, in an ascomycetes to make the sexual structures. So So what are those genes look like in these species? And so and this is with Greg Jedd's lab in Singapore, who's a cell biologist, works a lot on some of these aspects of cell biology and in Neurospora especially. And so what we started with was we said we took organisms like Neurospora and others, and we asked what are the genes that are shared among them, among organisms that we would call complex multicellular. So there's lots of genes in the genome. I'm going to walk through a couple of these plots. This one is a set of genes that we know are important in plugging holes in the septa. Okay, and I'm pretty sure I put the figure of what this looks like in a second, but these are a bunch of proteins that we know are important in separating cells. And we think that separating two cell types is probably an important part of that, of being multicellular. And so only those genes are only found in these ascomycetes, they're not found in anything else. And then there are other genes that are involved in what's called the septal pore cap, which is another kind of plugging technology or component that fungi use, again, to gate their cells. And the way that we think about this is you're a submarine and you've got different compartments, and you get you're in a lab and someone brings a razor blade and cuts part of your hyphae and you start to bleed out. Okay, if you're in the submarine, you'd like to close off those doors and stop this cytoplasmic leakage. And so there are these pores that can be occluded by a protein that prevent that leakage. And the proteins that are involved in blocking those pores in ascomycetes, in Physizomycotina ascomycetes, are these proteins like hex and lectin and whisk. Okay, and there's a different set of proteins that are involved in that pore in basidiomycetes here. They're so independent origins of that technology or that innovation. And then these proteins here are involved in hyphal fusion, so a lot of these do not seem to be found in NEOLECTA. So basically, dead end on this is not necessarily showing us a component that's related to multicellularity from our hypothesis, sort of starting out hypothesis. And so let me just visibly show you again. In Physizomycotina, you have this sort of raft, this lifeboat raft. I must be scared of drowning, I guess, I all these examples here, but you have this raft, and as there's flow through there, they get liberated and they plug the hole. Okay, in Physizomycotina, and there's a septal pore cap that's involved in that. In basidiomycetes, it's a little bit less well known. We wanted to know what does it look like in NEOLECTA. This is what it looks like under the EM of that pore in NEOLECTA. So it actually makes this protein aggregate, which is involved in plugging, and I'm not showing you the picture of this in Physizomycotina, but it's similar but different in the sense that there's some sort of protein aggregate that's fitting in that hole, but it's not the proteins that we know about. It's something else novel that's gone in there, and we don't know what it is yet. We've tried a couple of different things. We don't actually have the protein yet, but visibly you can see that it's doing that. And so again, just reiterating what I just said, but this is what's known in Physizomycotina. There's this hex protein that gets decorated with these sticky things that stick together, and they're involved in plugging in that hole. And all of those proteins are necessary for that, and they're not found in, they're only found in those Physizomycotina fungi. Okay, so that's cool, because in a way we further confirmed that this is a really unique innovation that the Physizomycotina evolved to deal with how they plug their septa. So that is kind of cool. It's not something that's older than the Physizomycotina. But we can look at other proteins, so that I'm sort of taking you down this big picture, and there are other proteins that do seem to be shared among many of these, but not necessarily specific to the complex multicellularity. And there are some proteins that are involved in really old, the stripac complex is a really old complex. It's actually found in lots of eukaryotes that it's involved in development. And so we're not surprised that it's basically found to be conserved in a bunch of these. But basically our candidate gene approach didn't necessarily pick out totally obvious things that we were hoping that would be able to tell us what's going on from there. But we did use a different approach where we say, can we find genes that really follow the pattern and that really tend to be in the complex multicellular fungi and missing in things like Saccharomyces or Candida, which are single celled. And we do look at things involved in light response. Reactive oxygen species, again, also seems to be quite, not completely specific to, but quite enriched and missing in the Saccharomyces lineages that relate to reactive oxygen species. And some of the genes, in terms of some of the chitin synthase genes involved in remodeling, are only found in these species that have the complex multicellular. It's not a perfect match, but it is it is useful to look at that. And so here's an example of a protein that's a septal-pore associated protein that is, for example, female sterile. So it's probably a protein involved in making a sexual structure in, this is in Neurospora, where we're able to do the experiments, and we can show that that's an important protein that has this pattern of being sort of only in these two fungi and not in the others. Okay, so we're trying to get at more than just making gene lists. We're actually trying to test these, but we have to do it in Neurospora because that's a system where we can knock out the genes. And so this is a sort of a further narrow down to novel genes that seem to show this pattern. So again, you have to squint a little bit. We obviously use this sort of darker color to represent kind of degree of conservation. And so some of these proteins may be not necessarily all lost in the single-celled fungi, although some of them are, but that we start to test. These are genes that have not been necessary. We don't know the function of them, and but they seem to show a pattern that's consistent with this complex multicellularity. And so what Greg's lab did is tag these and look at where they localize to. And for example, some of these proteins the details aren't so important, but that we can look at where they localize to and see that many of them are enriched for transmembrane domains, and some of these have novel localization or a novelly associated protein with mitochondria. So just the take home is, this is more of like a framework for it, is to try to use this approach to prioritize genes that might be important in that process. And so one final kind of example to walk through from the work was that there was this this protein that's involved in this diene binding, and this P150 glued protein is involved in this microtubule binding. And so what's interesting is this is a protein. What I want you to get away from this is it's not about whether or not the gene is present or absent. What's interesting about this, and this is what this plot is showing you, is that there are parts of the protein which are shared among the fungi that have complex multicellularity, and there are parts of the protein that are missing in the fungi that are single-celled. So they still have the same gene. So if you just go by orthology, you can find it. But that on par, most of the species that don't make these complex structures have lost parts of this protein. That's basically the take home of this figure and this figure. And so the dotted lines are the places where it's missing, and those are involved in some of the interactions. And so it might be that, again, when you think about this, we have a hyphae, and so some of the ideas, you have to transport things along a hyphae, and so that's part of the need for some of these binding and these proteins, the function that these proteins play. And so I think that's a harder thing to write really good software to be able to find, right? This was an effort of really studying the protein and knowing what it did and knowing what parts of it were important in order to find a pattern like this. So, I mean, it makes our job harder. We can't just sequence a genome and say the genes that are in the complex ones and not in the single-celled ones are the answer, right? So these are examples of things where I think as we dig into it, we can find a better interpretation of the important parts. Okay, so and there's another way to look at that picture of that same bit. So there are bits that are missing in some of these single-celled ones, but that where the NEOLECTA protein and its sister species really still have it, but then it starts to get lost again and shrink in some of these single-celled ones. Okay, so you may not be convinced that this is exactly what's happening, but I think these are the trends that we're trying to dig into deeper instead of just looking at presence absence as a way to make a characterization. Okay, so again, we can look for gene loss across a big set of species or gene gain. And so, for example, I didn't show you the data, but we know that flagella were lost in species that don't have flagella, and there are early lineages that do have flagella. That's a very easy signal to find. We can find those proteins missing in all the species that don't make flagella, but we can look for other characteristics that relate to things like multicellularity. And here we've got this more nuanced way of looking at it, but we do think that we have some tools for being able to jump into that. Okay, so in conclusion, this is the Wild West of some of our work, but some of our the ZygoLife project team is a big collaboration of a lot of different labs that have been contributing to this, and students in my lab that have been working on different parts of this, and postdoc Usmani, who did all the NEOLECTA work in my group. And I think there's the color version of that. With that, I will take, and also take your kid sampling in the desert. It's always fun to do. He wasn't going to smile for the camera, but it was good. So with that, I will stop, take any questions if there's any time, and thank you for your attention.
Thank you so much for your talk on the best organisms on the planet. I think it was a great overview of phylogeny, function, and evolution. And thank you so much. In commemoration and in gratitude, we have a little plaque.
Thank you.
For the lecture series. Any questions?
Yeah. So in your analysis of the trying to infer genome duplications, how can you separate that from possible hybridization events?
Yeah, that's a great question. I guess we would have to do a secondary set of analyses to try to look at so I think we should be sequencing more than one individual for any given species, and that would allow us to at least add some population variation in that to see if the same region is, oh, you're thinking like a little bit older hybridization. So sure, I mean, the mechanism by which you duplicated maybe doesn't have to be an endoduplication. It could be a hybridization that driven, I think it's more, are there twice as many genes is the first signature. And then, you know, it depends on whether you're going to maintain those duplicates. So we do see in some species what looks to be diploids that then lose heterozygosity, and other kind of patterns that really suggest that you kind of, it's really a diploid that does that hybridization, as opposed to really like maintaining a duplicate set of chromosomes. So I think that's probably part of what we might have to tease out as to how different the alleles are in those duplicates to really get at it. But you're right, there's a gradient. Yeah.
There's a feature in rust fungi called obligate biotropy. Is it possible to understand how those genes became dedicated to maintaining that system?
Yeah, that's a great question. I mean, I think the answer is hopefully yes. We'll be able to get at the either the metabolism requirements that have made them obligate, or that support that obligate nature, or the best way to be able to have it is you have a species that isn't obligate, and to be able to compare what's different, at least at that starting point. So I guess that's where we would start from. But I don't know that we have. I haven't focused on that lineage yet. There are a lot. The rust genomes tend to be pretty big, so which is interesting, right? They're obligate, but they're not necessarily like reduced. And so I think trying to understand some of that is still ongoing as people get more sampling of it. So
Lots of retrotransposons.
I think so.
These extremophile co-adapted fungi, do they represent taxonomic diversity of fungi in general, or is this just one narrow group of highly specialized organisms?
So our extremophiles just in one part of the tree, basically, so there's a lot in the Deuthidiomycetes that represent that. There are other examples of extremophiles, depending. I mean, thermophilic extremophiles live in or are found in different parts of the tree as well. The cold ones. So we've done a microbiome ITS kind of study, and we do have. What's interesting is you get a lot of lichens out of that, because that's what typically is the biomass inside those. But when you go to do culturing, you can obviously see a bit more of the species. Most of the ones that, so we do have one Basidiomycete or Rhodotorula that seems to be associated with the rocks, but then it's predominantly Deuthidiomycetes that have been put in the culture collections that we're able to use, or we've been able to kind of work on. And I think there are some other Pizizomycotina, other filamentous ascomycetes. So the K3 ALEs would be the other group where you see those as well, which is another. There's sort of two groups of kind of black yeasts, and that's kind of the other group. Chelsea?
So on the multicellularity question, and I'm sort of thinking about the protein plugs, but this would be various traits that are associated with multicellularity. How many times do you have multicellularity evolving?
Mm-hmm.
Is it because what I was talking about the plugs, and you've got 3 different types of plugs
That's
happening. Right. Are they using an ancestral mechanism, or are they just all doing it differently?
Well, I think so. That's a great question. I think how many times multicellularity evolved in fungi is somewhat debate as to whether or not what you define as complex multicellular. So I mean, just forming any kind of complex fruiting body probably might be multicellular. So we have to. So that's that's a debate. But let's talk about this very specific thing, the plugging. You need septa to be able to have the plugging. So we don't think that the ancestor of the fungi had it. We think that probably, at least from our reconstruction, you know, that we do see septa in some of the what's called the GENOMES, but within the zygomycetes, we do see one other example of septa, but we haven't, we don't know anything about the pore structure there. So I don't anyways. But because of this work at NEOLECTA, we can at least say there's 3 origins of that kind of plugging in the examples that we've looked at there, which is more than before when we would have just said 2. And previously we'd say that the dicaria fungi, so the ancestor of the ascomycetes and basidiomycetes, must not have had that because we independently evolved it 2 different ways. So that might mean that the ancestor of dicaria didn't really have septa or true septal, need for true septal pores until later because of that. I mean, that's the parsimonious interpretation of that, unless there's some gain loss thing that we, you know, it's really rapid.
So with the genomes, then you would see there's something associated with needing to plug your septa, but it could be different in every single case, and you wouldn't see loss necessarily. Are there any cases where you go back to single cellular from multicellular?
Yeah, I think so. I think that so I think that Safrina-Mycotina probably represent that, and that you see simplification. And we think Saccharomycotina, if you look at the tree, Saccharomycotina represents a simplification too. So probably the ancestor of Saccharomycotina and Saccharomyces was more complex, and that you've seen simplification or loss. The genomes are smaller, and they have fewer cell wall encoding genes, a lot of things that might be involved in that process. They don't have these hyphal fusion genes that are shared. So I think that, yeah, we do think that it's actually simplification, or however you want to do, you know, that sort of has a loaded term, but like losses are driving some of that. But yeah, there's one other point, but that was that, yeah. Yeah, Alan.
I'm assuming there's a wealth of data, genetic data, with model Neurospora,
Yeah.
model filamentous fungi.
Yes.
How well does that, do those data inform phylogenetic analysis?
Well, I mean, in one part, we are still probably working on integration of those data in the right way. So there's been sort of siloed, you know, there's a really good Saccharomyces database and ability to look at gene function in those, and we can map those onto a Neurospora gene and ask, is there good evidence they have the same function, or they're part of the same process? Although sometimes that's a circular inference because the annotation in Neurospora might be driven from the Saccharomyces homolog. But in terms of how it maps on the phylum, I don't know that we've really looked at it in terms of phylogenetically. I think we've just tried to ask, are there consistency between them? Probably the best place where it's been done is like there's a systematic knockout set in Pombi and in Saccharomyces, right? And so that's a case where you'd want to ask for each ortholog, is the phenotype pretty similar or not? And in Neurospora, we have a systematic knockout collection, but they've not been all phenotyped. And so probably about 1,500 to maybe about 2,000 have really been phenotyped out of that 10,000 knockout collection. So I think that's what's limiting it right now, is really to systematically have that. So people are doing like growth rate and sexual reproduction and a few other things are being scored, but I wouldn't say it's actually, the resources are there, but I'm not sure all of it's been, you know, kind of really assayed. So, yeah.
In all this genomic data that you've kind of been presenting on today, one thing that I kind of thought was missing was sort of the mitochondrial DNA. And do you have any insights on kind of the evolution of mitochondria within fungi? Or have you looked at that at all?
Right. So the short answer is no, I have not looked at that. There are a couple of labs that have been focused on mitochondrial evolution, more focused on some of the chytrids and the zygomycete fungi than others. So it's definitely an open question, I guess. The short answer is it needs better work. I think there's just not enough people who've really delved into it. Sometimes with the genome assemblies, it takes a little extra effort to pool the mitochondrial reads and call it that assembly. It's not actually that hard, but it hasn't probably been done as systematically as it should be. So I think it's kind of an open space that hasn't gotten as much attention.
I guess, do you think any of that information could inform some of the phylogenetic analyses and help? Because I know, you know, animals, they use a lot of like cytochromes, subunits, phylogenetic analysis. I don't know.
Yeah.
And some of those gene subsets that would change the picture of the way, I don't know.
Right. It's a good question. I mean, I don't know if, inheritance of mitochondria is an interesting question, and sort of which parent donates in, and the interactions between the nuclear genome and the mitochondrial genome there definitely are interesting experiments, mostly in like multinucleate basidiomycetes and sort of tracking things through. So there are interesting research questions about those interactions, but I don't know that like a systematic, here's all the mitochondria and their content and what's been gained and lost. There's sort of like individual examples of like intron gain and loss in some of those, and yeah, I think it's an open space that someone needs more work. So, yeah.
Thank you very much.
Thank you very much.
This has been a production of Cornell University, on the web at cornell.edu.